Hey everyone, so welcome to a talk about one of my favorite topics, surprisingly enough, which is the chemistry of biology, otherwise known as biochemistry. So we're going to review some basic and not so basic chemistry concepts. I'm hoping that a lot of what we are going to talk about is in your brain somewhere as an ancient memory, perhaps from your courses that you took in this subject, right? You had to take chemistry to get to microbiology, so some of it is going to sound familiar. And then we're going to talk a little bit about how bacteria take chemistry and do some stuff with it, which makes it all that more interesting and makes them, once again, have those superpowers. So I'm going to start slow, kind of go back over some of the basics, but there's a lot of basic chemistry, which the chapter in the textbook does a very nice job reviewing, which I suggest if it's been a while, you might want to go do that. I'm just going to focus on how chemistry relates to what we need to know here in microbiology. So, just kind of starting with the basics of what is matter, right? Does matter matter? I always wondered that. All right, so where did they come up with this word? So matter is defined as anything that takes up space and has a mass. So essentially, that's pretty large. Everything is matter, which means also that molecules that cells are made out of, the tissues that are made out of cells, organisms that are made out of tissues, we're all matter. So what's important to know about matter is because matter is composed of molecules. Molecules are combinations of atoms. Atoms are held together in molecules by bonds. There are many different types of bonds, which I won't review, but just realize that when atoms come together and form bonds, that energy is stored in that matter. And generally, the larger the molecule, the larger the matter, the more energy is stored in that matter. So with that in mind, that's where we're going to kind of go with this. So matter stores chemicals that can be used, organic chemicals that can be used for building blocks, and it also stores energy. Biomass is matter that's made out of biomolecules. So this is biology is what can be considered biomass. That maybe a better word would be biomatter, but nobody calls it that, so we're going to call it biomass. So the definition is the mass of all the organisms that are found in a community of organisms, which is also a very broad definition. But realize that biomass is matter made out of biomolecules, which in the days of chemistry, they called those organic chemical compounds, meaning they're made out of carbon and hydrogen at least, and often contain hydrogen and other atoms, other elements. But they are organic because they contain carbon and hydrogen. And so life on this planet, as far as we know, at least at this point, is organic, that it is constructed out of molecules made of atoms, which include at least carbon and hydrogen. So hopefully also you remember that the types of biomolecules that biological matter is made out of, there are 4 major classes of these. So there are the lipids, the nucleic acids, the carbohydrates, and the proteins. And in your previous classes, you spend a lot of time learning about what those are made out of, what function or role they play inside of cells. So you want to kind of remember what they are composed of, right? Lipids, are made out of smaller biomolecules, which many refer to as metabolites. Metabolite refers to the fact that these are compounds that can be used to construct larger molecules from, or they are released when those larger molecules are deconstructed. So the lipids, for example, the metabolite are various types of fatty acids and then other compounds, but we'll just stick with the fatty acids for now. The nucleic acids, the smaller metabolites, are the nucleotides, of which there are 4 types that come together to form these very large molecules called macromolecules. Carbohydrates, a broad class. Again, there are simple carbohydrates and complex carbohydrates. They are the monomer, or the metabolite would be simple sugars, right? So just those single ring sugars would be what these are composed out of. And then the proteins, which the metabolite are the amino acids. So amino acids go into making proteins, and they are released when proteins are broken down. So remember those, because you will see that as we talk about, we put this all in a larger context, we're going to go both ways with these. We're going to build biomatter, and we are going to break it down in the processes which are collectively referred to as metabolism. So before we do that, let's just kind of go look at another very basic concept of both physics and chemistry about matter, which is matter again, it's a storage molecule. These are molecules that store both elements, and they also store energy. Remember the first law of thermodynamics, which said that matter and energy are conserved in chemical reactions, meaning that whatever you start with comes out at the other end. It may not be exactly in the same form, but there will be the same amount of matter and energy as at the end of a chemical reaction as what went into it. So let's look at that. So we're going to use as our matter a gummy bear, which you can see here on the screen. This is a gummy bear. We are going to do a chemical reaction on this gummy bear where we are going to transform the matter and the energy that are found in this gummy bear. So what is the matter? We have, if you've ever looked at a label of what gummies are made out of, they're made out of sugar, obviously, sucrose, and realize that a molecule, one molecule, and I would bet there's lots of molecules of sucrose in that gummy bear. One molecule holds 3.94 kilograms of energy per gram. And then protein molecules have gelatin in particular, because it was molded to look like this gummy bear, have a little bit less than that, right? 3.35 kilocalories. That's a unit of energy. Okay, so there's lots of molecules of both of these things in that gummy, probably some other stuff too, red dye number, whatever, and other things. We'll just kind of stick with the matter. So what are sugar molecules made out of? They're made out of carbon, hydrogen, and oxygen. Protein molecules are too, with a few other things, right? There's a little bit of sulfur thrown in there. There might be others. But the point being, when we transform this gummy bear, so we're going to do a chemical reaction. We're going to add a chemical catalyst to this, and heat, a physical catalyst, for this reaction to occur. When we do it, you're going to watch in real time the transformation of this gummy bear into other forms of matter and energy. So your job is, while we watch this, to try to figure out, all right, so if we're transforming, not destroying or making energy, then where is this energy going when we put the gummy bear in the chemical under heat, and where is the energy going? Okay, so let's watch this gummy bear. We're gonna pick him up and put him in a test tube with some hot potassium permanganate, and you'll see what happens to him. Yikes! Can you hear him screaming? I always feel sorry for the gummy bear. The gummy bear is totally innocent, but you know, there are no humans that would volunteer for this experiment, so the gummy bear stood in for us. So as you watch that, we put the gummy into the heated chemical solution, which caused it to break down. So the carbon, the hydrogen, and the oxygen that were in the molecules of sugar and protein, where did they go? So you probably noticed that while the reaction was occurring, you could see steam, right, coming out at high speed. That's water, H2O. So there's the hydrogen and the oxygen. And it's kind of hard to see, but down at the bottom, you saw some black bits and spewing out of the top, and then you can actually see some black bits at the bottom, and that's the carbon. So the carbon stayed in the tube, but the other atoms, the other part of the matter, actually left as gases. So then what about the energy? So as you're watching this, you probably noted that there was flames spewing out of the top. If we had sound associated with it, you would hear all this crackling and popping noises that were coming out of that. And there were flashes of light and there's heat. So the energy is being transformed into heat energy, light energy, and sound energy. So that's where it went at the end of this reaction. So whatever energy was in that gummy bear was released in a sort of dramatic and semi-explosive fashion. So that's what conservation of energy refers to. And you could, as we did here, do it chemically, but realize that in biology, if I had put a real biological thing into that test tube, it wouldn't have been very good for the living thing, because these types of chemical reactions can be quite destructive. So what biology does is slow it down just a little bit. But that means we need to talk about the kinds of reactions that occur when we are transforming energy in biology. And for the most part, those are oxidation and reduction reactions, which are called redox collectively because you can never have one without the other. And that is because it is a conservation of energy issue, once again. So oxidation and reduction reactions, probably you remember what those are because you had to do this quite a bit in chemistry, where you have an electron donor, something that has electrons, an abundance of them, and it transfers them, it gives them up, it loses them, if you will, to an electron acceptor. All right, so in our example here, A is the electron donor. See the nice juicy electrons that are associated with whatever A is. And B is going to be the electron acceptor. And that sounds like a very benign relationship, but what I'd like you to realize is that the electron acceptor wants those electrons, wants them more than A does. So it's not exactly like, oh, here, take my electrons. It's more like B saying, give me those electrons. I want the electrons. And then if some other electron acceptor comes along that wants those electrons more than B did, then B is going to give them up to the next thing and the next thing and the next thing. So the electrons can be transferred right down a pathway, which we'll see in a moment. So the electron donor A gives up those electrons to B, which wants them more. And when that happens, A is oxidized because oxidation is a loss of electrons. So the electrons are lost from A, so A is oxidized. And B accepts them or steals them, however you want to look at that, and B becomes reduced. So you did oxidation reduction reactions often probably with metals because metal elements that are considered metals do this all the time. It's like, here, take my electrons, give me those electrons. They are known for that sort of thing. And in biology, this happens also, but just to simplify it a bit, the oxidation and reduction reactions often involve electrons that are part of hydrogen atoms. So looking at the structure of a hydrogen atom very simply, there's a proton in it and there's an electron in it. And so as opposed to just transferring those naked electrons, if you want to look at it that way, it's a transfer of hydrogen atoms that have those electrons associated with them. So chemically speaking, what we can look at, this is a biological redox reaction. This is our electron donor, and for fun, I'll give it a name. It's lactic acid or lactate, and that's going to be our electron donor. So you'll notice right here, there's a hydrogen, and when it comes in contact with that electron acceptor, which by the way, wants the electrons more than this organic compound does. So this is nicotinamide adenine dinucleotide called NAD, which is a coenzyme. It is a nucleotide that acts as a helper, if you will, to a chemical reaction, a chemical redox reaction, where the job of this thing is to just hold electrons, hold them for a short time, so it will accept them, but it wants to give them away almost as quickly as it accepts them. So it's a transfer molecule, it's a carrier, if you will, electron carrier. So that's going to be our electron acceptor. So lactate gets oxidized, forming, this is pyruvic acid, by the way, if you wanted to know the name of that organic compound. So lactate is oxidized to form pyruvate, and this electron, which is here with this hydrogen, is transferred to NAD, and NADH is produced chemically. So NAD accepts the electrons from the lactate, and it becomes the reduced form of NAD, which is NADH, written NADH. So now NADH is carrying those electrons, which, as I said, as soon as it encounters something else that wants those electrons more, it's going to give them up. And that forms the basis of electron transport chains. And we'll talk about that when we start to talk about metabolic pathways. So redox, transformation of matter, right? So here's the transformation of matter. Realize that the transfer of electrons is also a transformation of energy. So the energy that was held in this bond just got transferred here. And you'll see many examples of that with other coenzymes for these kinds of reactions. Okay, so just to talk about another thing that's very important in biology, another type of chemical thing that's very important in biology, and that is water. So if you pay any attention to research and what, looking at astrophysics and life on other planets, is there life on other planets? One thing that most people most of those types of scientists look for is there any chance that there's water molecules on that planet, because water is so critically important to life? To which I always want to say, on this planet. But anyway, I'm not an astrobiologist, so we'll let those guys figure it out. So the thing is, what's so great about water, right? So in my opinion, if you are ever asked a question on a philosophy exam where it's an essay, and the essay is, what is life? And they expect you to write 14 paragraphs on, all the existential reasons why life is profound and different. You can get an A on that exam by simply answering, life is hydrogen bonds. Because really, that's what life on this planet, anyway, is. The hydrogen bonds are the basis for so many of the molecules that form the cells, and all the interactions between things that occur in cells are due to this unique property of water. Okay, so what is that property? Water is made out of one atom of oxygen and 2 molecules of hydrogen. And it is a relationship where oxygen and hydrogen share electrons. So it isn't a transfer of electrons, it's sharing them. So these are covalent bonds, if you want to remember those terms. But the way this happens, because there's 2 electrons pairs to be shared, oxygen will hook up with 2 hydrogens. And that, because oxygen is much larger and has more electrons to begin with, this is a more negative end of a water molecule. So the molecule, the H2O molecule, has a negative end to it, and then it has these 2 positive poles or terminals. So water is considered polar, meaning an opposite end, right? So we've got the negative end, and we've got these positive ends. And we all know that if a positive and a negative get close enough together, what do? They're attracted to one another. So they're held together in an electrical fashion. And that is the basis for a hydrogen bond, which is actually not really a bond. It's a force called a van der Waals force. But it's kind of like this negative side likes that positive side. So they hang out together. And it's a fairly strong force. So it's not a bond where, a huge amount of energy needs to go into breaking it. It is just like, hey, I like you, I'm going to hang out with you. So these 2 hydrogen or water molecules are held together by a hydrogen bond. And as you might expect, in a drop of water or a glass of water or a body of water, there are bazillions of water molecules, and they are all sort of held together with these hydrogen bonds that are being made and broken and made and broken all the time. And this is under the influence of heat, right? So water is also one of those things that can exist simultaneously in 3 states, the state of the liquid, solid, and gas stage, which is also another one of those fun things. But nonetheless, the hydrogen bonds in biology play a very important role. So without getting into too much more detail, let's just look at why, those hydrogen bonds are important here. So first of all, water, because it is polar, water molecules will interact with other polar compounds. And by compounds, I'm talking about molecule, right? Something made of more than one atom, combinations of atoms. So to be polar means you have two oppositely charged ends. So water has a positive and a negative. And when water comes in contact with something like, in this example, sodium chloride or salt, because this also, right, so our little sodium has a positive charge and our larger chlorine has a negative charge. So they hang out together for the same reason that, other positive and negative things do. So this is called an ionic bond because it's a fairly strong bond. So these guys are held together. They'll form crystals, right? That's where salt crystals come from. So if there's no water around, the salt molecules will form large crystals and they'll stay there in solid form right up until water shows up. Because when water shows up, remember, they also have positive and negative ends. And so what happens is our polar compound, our ionically bonded compound, dissolves, it separates. So our sodium and our chloride will be broken apart. They'll each be surrounded by water and there they go. So it's called dissolving, but this is how solutions are produced. This is the basis for things like pH, for making solutions, for actually, a lot of what goes on inside our bodies are solution-based chemistry, meaning that there are chemical reactions that occur because there is water present. So polar compounds are called hydrophilic. Philic means to love. So these are water-loving because they react with and dissolve in water. So the opposite of that would be nonpolar compounds. So these are compounds that don't have opposite ends. These tend to be things that have lots of carbons and hydrogens, so all sides of the molecule are pretty much the same. So shown here is a fatty acid, right? These are fatty acid molecules, which are, each of these little bulges is it's a long chain of carbons with hydrogens off of them. So there's no charges that way. So when you take a nonpolar compound and put it in water, there's no reaction going on. And what happens is the hydrogen bonds are still being formed between the water molecules, and they drive the nonpolar or hydrophobic water-fearing compounds to bundle up together. So they kind of get surrounded and they end up bunching together. They don't bond to each other because they are perfectly happy bonded to themselves. So they just sort of hang out surrounded by water, which basically says you stay there because you're not going to react. So sometimes this forms layers, right? So you can see layers of nonpolar things like fat. When you add fat to water, the fat will sit right on top, cream on milk, right? That kind of thing. But in biology, why this is significant is because at some point in the history of life on Earth, before there was probably life, when there was just chemistry, when there were just chemicals, nature formed compounds that were organic, then they formed lipids. They added phospholipids, right? Put phosphate ends to those lipids. The phosphate end actually does carry a charge attached to fatty acid ends, which don't. And so what you have are these compounds that one side reacts with the water and the other doesn't. And you can see what happens. So here is something called a bilayer of these phospholipid molecules. So the phosphate or water-loving end react with the water, so they're pointing out, and the hydrophobic innards, which are fatty acids, are protected on the inside. So you know how when you throw fat in water, you get that nice single layer. With phospholipids, you get bilayers. So this is a cell membrane, which hopefully you remember, a bilayer of phospholipid molecules. The only reason that membrane stays together at all is because of water. So the invention was, right, let's take these structures. So this is something called a liposome. This is a micelle, so you can see it's a monolayer, if you will, one layer. But obviously this is where cell membranes came from, because these things formed, and for whatever reason, the other molecules decided, hey, you know, it'd be cool if we were inside of that particular structure. So water is what actually produces that. And then, you know, just a shout out to cell membranes. If you remember, cell membranes, I consider one of the essential must-haves to be a cell, because without a cell membrane, there's no insides or outsides. It's, you know, so I think to be a cell, the essential structure is actually one of these things. And so here's a representation of a cell membrane. It is a perfect structure to do this because it is semi-permeable. Some things can move freely back and forth across it through diffusion. Some things, like water, requires osmosis. Larger things need transport, which of course that requires varying degrees of energy. And so, you know, biology built on all of this. So from our very simple movement of things back and forth across that membrane, from there, the addition of we need pumps to do things and being able to channel the passage of molecules across this membrane and create potentials, meaning potentials to produce energy across membranes, is one of the hallmarks of life. So water did that for us. So thank you very much, water. That's why we talk about water in biology all the time. And here's something. So we've known about most of this stuff. We've known about DNA for a long time, that the reason why DNA is a double helix is not just because of the bonds that form between individual nucleotides that form those long chains, and then, you know, the chains wind up to form the double helix. Water is what drives them, keeps them in that position. So notice that this paper was from a year ago in 2019. And finally getting around to doing these experiments because we have the technology that allows them to, but realize that it's not just about creating the double helix and keeping it stable. Water does that, but it's also about the fact that the cell can control how it unwinds and winds up the DNA. As soon as it wants to do something with the DNA, the cell will flip the DNA molecule so that it is exposed to the hydrophobic environment. So that means there's a reaction with and a repulsion by water, which is this, you know, the hallmark of DNA, which is of course the other thing that makes a cell, which is a genome made out of DNA. So, and you can't see it because I put my face right over. I don't actually know where my face is on your picture, but this is NASA's searching for alien life is looking at this. So it's not just about, oh, that's really interesting that water makes DNA do what it does for us, which is to get copied and to be transferred into protein or whatever. It's also something that we're looking for when it does come to astrobiology, which is, come on, you gotta admit, that's pretty cool. Okay, so enough about water. Let's get back to energy and talk about some of the chemical reactions that we're talking about here. Okay, so back to transforming matter and energy. There's the chemical way, which is probably how this all started way back in the beginning. I'm sure there was a lot of this sort of thing, where notice in this case, we have a sugar molecule, right? That's glucose right there. And you can combust it, which is to break it down with oxygen if you wanted to, doing it the chemical way. And if you did it this way, if you did it with heat, for example, what would happen is once you added enough energy through the heat or the chemical reaction to overcome the activation energy, notice what happens. Boom. All that energy, I always do that. I always hit the thing before I want to. All that energy is released in one big bang, literally. So I envisioned it to be like this, right? So we go from enough heat to hit the peak of activation energy, and then look what happens. Boom, we have a nuclear reaction. The sugar is broken down. Here's the matter. And then all that energy is released. How is it released? As heat or light or something explosive, which is useful for some things, right? So if you want your car to run, it's useful to have this kind of reaction occurring because that energy gets released and then utilized to turn the wheels of the car, right? But for biology, maybe this isn't exactly the best idea. So imagine every time you ate a gummy bear, you exploded it. That I don't think that would work out very well for any of us, right? So there's the chemical way, releasing that energy abruptly, and there are uses for that. But looking at the biological way, so as opposed to releasing it all at once and abruptly, what biology does is to release it a little bit at a time. So small activation energies, small releases of energy at several steps. So that forms the basis for these chemical steps or pathways. And this is how biology transforms matter and energy, is a little bit at a time, kind of like this guy, right? So he's just taking it slow, not blowing anything up, trying to get whatever it is that this guy's holding. And the good news is, so the transformation of matter is exactly the same. Carbon dioxide and water are what the sugar molecule is converted into. But the energy, because it's being released in a controllable fashion, can be transferred and stored as a storage molecule. And in the case of biology, that storage molecule is this one, which is called ATP, adenosine triphosphate. It is a nucleotide, and it's called triphosphate because there are 3 phosphate groups. And realize that forming each one of these phosphate bonds that hold these phosphate groups together requires energy to go in. So the stored energy is held between those phosphate groups. And of course, the energy that goes into making those bonds can be released later on. So it is the storage molecule for energy in cells, so that we don't have to keep blowing things up all the time. It's a brilliant system. So that forms the basis, these kinds of reactions form the basis for cellular metabolism. So metabolism is a word that encompasses all the different types of chemical reactions that occur in cells. And there are thousands and thousands of chemical reactions that occur in cells. So collectively, it's metabolism. Realize that there are kind of 2 arms to this. There are catabolic reactions or catabolism, where the biomolecules, because that's what we're talking about here, they're being broken down through sequential oxidation reactions, through pathways of oxidation reactions. So they're being broken down to metabolites. And as they are being broken down, so here's our organic compound, right, our biomolecule. As it's being broken down, the large biomolecules are being broken down into the metabolites. There they are. And that releases energy. So energy is being liberated, going from a large thing with lots of bonds into smaller biomolecules. And as those bonds are broken, that energy is being transferred to ATP. The other half of that is anabolism, which is the biomolecules, the smaller ones, the metabolites, are being combined, and that requires energy. It requires energy to make those bonds. Here we're breaking bonds. To make those bonds, we have to add energy in. So in this case, energy and metabolites are being consumed, and when you pair them, when you pair catabolism with metabolism, you can see that this becomes cyclical. And so as long as there is a supply of metabolites and energy, you can make biomolecules. As long as there are biomolecules, you can make metabolites and energy. And that's how cells function. So I want to show you, this is what the inside of a cell looks like in terms of its metabolism. All these many different types of chemical reactions are occurring. Okay, so each one of these different colored lines with dots, each dot is a chemical reaction. Each line is a reaction in between. So this would be one metabolite to another to another. This is something called the Krebs cycle that you see here. This is carbohydrate metabolism. So in other words, breaking down carbohydrates, this is a way to release things that then go into amino acid metabolism, that go into energy metabolism. So this would be making ATP. Here we're going into making lipids. then the amino acids go into metabolism of other amino acids. So it's this huge interconnective highway of all of these different chemical reactions that are all dependent on each other for the net outcome, which is metabolism, making biomolecules, using energy, breaking them down to get energy back. So this is kind of how I envision what's going on inside your cells right now. So you can sit and watch it and just be like, oh, yep, there's the Krebs cycle. And over here, you know, we're going to be making some lipid molecules and fatty acid molecules over here, which of course are going to go over here where we're going to need them to make fats, which is what these guys are doing. And, you know, you can get lost in watching that. And if you want to go back and watch it without hearing anything and just sit there and watch it, which is fun, you can just mute me and stay here for the rest of the time because it is fun to watch. But envision that occurring in each and every one of your cells to varying degrees for the whole duration of your entire life. So that's cells in general, prokaryotic and eukaryotic cells. And of course, because this is microbiology and I am who I am, which is I will gladly admit that I am a bacterial bigot. So we're going to talk about prokaryotes. We're going to talk about catabolism and a little bit about anabolism because we need to in prokaryotes, meaning bacteria and archaea. So a lot of what we know about this comes from our studies on bacteria, meaning the big B bacteria, and the ones that we can grow in culture. So there's a lot about bacterial metabolism we still don't know and we are working on, but so we'll just talk about the stuff that we are able to know. Catabolic pathways, okay, so these are the ones that take biomolecules, break them down, release energy, release metabolites. So the way that the pathways that are available to bacteria, prokaryotes, to do this, they have a variety of ways to do this. So one way is through, if it is a sugar that to be broken down, right? So carbohydrate catabolism or sugar catabolism. The pathways are glycolysis and fermentation. And if it is something else, then we have Krebs cycle and the electron transport system. And I just want you to realize that fermentation as a pathway and respiration as a pathway are complete pathways. Glycolysis and the Krebs cycle are intermediate pathways, getting us to the end products that we're seeking, which is mostly biomolecules, the smaller metabolites to make biomolecules from, and then of course ATP. Okay, so in both glycolysis and fermentation, sugars are being oxidized. Sugars, notice, bold, underlined. It means the only thing, the only substrate that works in glycolysis, which means sugar splitting, are sugars. So you have to have sugars present for these pathways to even occur, okay? So sugars get oxidized, and remember we're talking about a redox pathway, so a redox chain, and pyruvate and other metabolites get reduced. So that's the electron acceptors that we're talking about here. These are the electron donors that we're talking about. So the energy, the transformation of energy goes to ATP for the most part. And what we end up with chemically are either alcohol or acid end products, which are chemicals that actually we can detect. And us humans have actually done quite a bit with these end products because we figured out early on that there were good things associated with the end products of bacterial fermentation. So then the Krebs cycle electron transport system, especially electron transport system, are respiration, which you can start with glycolysis to take the products of that into the Krebs cycle. Proteins and lipids can also serve as the biomolecules that enter into the Krebs cycle. And what serves as the electron acceptors, oxygen can, and that's the process known as aerobic respiration. And here's where I get to say the word anaerobic respiration. And realize that only bacteria and archaea are able to do this as a way to make a living. By living, I mean making ATP enough to survive on. And so as opposed to using oxygen as the final electron acceptor, where oxygen is reduced, water is produced, you remember that from your old days, They use other alternative types of electron acceptors that are not oxygen, nitrate, nitrite, sulfate, carbon dioxide, etc. And of course, the end products are not going to be water, they're going to be other stuff too. So because that's what bacteria do, we're going to end up talking about it, and you're going to love it as much as I do when we're all done with this. But let's start with sugar, because why wouldn't we want to start with sugar, right? We can't live without sugar. So let's start with glycolysis. And in your textbook, the OpenStax textbook, by the way, at the end, there's appendix of all these different types of reactions. So you can actually go look at every single chemical reaction in glycolysis in fermentation if you'd like to do that. Your textbook proper gives you a brief overview of it. That's the way I prefer to do it, is to consider it from the perspective of the transformation of matter and energy. So for glycolysis, the matter we're talking about is glucose or other simple sugars, like fructose can go into this. Other monosaccharides can be used, but it has to be a sugar. And then there are 5 chemical reactions, each one catalyzed by an enzyme. The enzymes make it a slow and steady process. Again, a transfer of energy at each step. So there's 5 chemical reactions, which are actually where energy is being invested into this process. And then after 5 reactions, there's another 5 reactions that occur, which are the payoff, right? So the energy goes in, but then at the end, you get more energy out because you made that initial investment. This is, when your retirement account pays out to you, which is more than the money that you put in, or at least one hopes so. So there's the transformation of matter. Is glucose is first transformed into this, this 6 carbon thing is transformed into 2 3 carbons things, and then the energy is sucked off those, leaving behind pyruvic acid, pyruvate, right? 3 carbon compounds with, don't forget there's hydrogens and oxygens there too. All right, so the matter, that's where the matter goes. It is transformed from glucose into pyruvic acid, 2 molecules of it. So 6 carbons equal. Where did the energy go? So the energy is in the hydrogens. And as those are being removed and all these chemical reactions are occurring, the hydrogens are being transferred in that reaction that I showed you, being transferred to NAD, forming NADH. And also a reaction called substrate level phosphorylation occurs, where the phosphate group you see here is directly transferred to make ATP. So the energy, you would think, because I know everybody knows what ATP is, it's cellular energy. So if I were to ask you just off the top of your head, looking at this, where's the energy? You'd say, oh, the energy is ATP. That's correct. The energy is also in NADH, that carrier molecule. And realize that NADH molecules are not particularly, I'm just going to say, they're not really happy. So they accepted these electrons, and now they're like, look, I got all these electrons. I don't want all these electrons. What am I going to do with them? So they're going to turn around and hand them off one more time. But in the meanwhile, glycolysis is not complete because we've got these NADH molecules left holding the bag. So it is not a complete pathway. There's still energy left. And we need to finish that transformation. And so that's where fermentation comes in. So if you are a yeast, or if you are many types of bacteria, or if you are certain types of human cells for a really brief period of time, you can actually complete this pathway. The cell can complete this pathway, and we call that fermentation. So what we do in fermentation is the electrons that, so that energy that is being held with NADH is transferred back to an organic receptor. And so this is the electron acceptor in this case. Okay, so in this pathway, here's glycolysis, here's our NADHs. Notice there's our pyruvic acid. So what happens to those electrons that are being carried or held by NADH is that NADH gives them back to the organic compound, forming something else. So in the ethanol pathway, the end product of the pathway is ethanol. So ethanol is alcohol. As you probably already know, it's not the only type of alcohol, but it turns out it's the only kind of alcohol that humans can consume and not kill themselves with. So it's, you know, one of the things that humans figured out pretty early in our history. It was that this chemistry was going on. They didn't know why or how until Louis Pasteur showed up, but it was an interesting, they were happy that it did, that it happened that way. So now this pathway is complete. So the end product is ethanol. The NADH is happy because it got rid of those electrons. All the energy is transformed. Everybody's stable and happy. Ethanol is the product. We have ATP. So the ATP is cellular energy the cell gets to use, and everybody's complete. So this is what yeast do. There are also bacteria that do this kind of fermentation. Another type of pathway is called the lactic acid or lactate pathway. Homolactic fermentation, lactic acid fermentation is what it's referred to because the end product of the pathway is lactic acid. So chemistry is different, right? This is an acid, this is an alcohol. So lactic acid is an acid that has it is not toxic. It can be consumed, and it in fact is the type of acid that is responsible for the fermentation or the transformation of milk to yogurt and yogurt to cheese and that sort of thing. So the chemistry is just a little bit different. Again, here's glycolysis. There's our pyruvate. Here's our unhappy NADHs, and the NADHs give them right back to pyruvate, and chemically speaking, we form this product. So as I mentioned, lactic acid fermentation is something that bacteria do. Oh yeah, human cells too. And just realize that when you learned about this in anatomy physiology, fermentation is what our muscle cells do when they run out of oxygen. So they will ferment, but they can only do it for something like 12 seconds, and then they die, which is why after a while people run out of ATP and their muscles stop working because they can no longer produce, they've run out of fuel and they can no longer produce energy. There's no more fermentation. So bacteria can make a living by doing this. Yeast can make a living by doing this type of fermentation. By that I mean they survive. They don't just do it as a last-ditch effort to keep themselves alive. They can survive on doing that alone. So these fermentation pathways, they are many and varied. Ethanol, obviously the end product is ethanol. Lactate, the end product is lactic acid. Get ready for this because the next slide is gonna be fun and interesting to look at. And I just wanted to show you all of these other different types of fermentation pathways that are done by bacteria. So you're probably thinking, oh my God, is this gonna be on the test? So here's what I would like you to take away from all this, all right? So here is glucose catabolism. From here to here is our glycolysis. So notice that, so each of these arrows represents a conversion. So those chemical conversions that occur are due to enzymes, again, redox pathways, redox transformation, there's energy being transformed. But let's just kind of look at, here's glycolysis. So notice headed this way is an alcohol fermentation. So there's our end product. Here's, again, the reversal of, we've made NADH here. Here's the reversal of that, conservation of energy. The bacteria, or the yeast that will make wine and beer for us, by the way, is Saccharomyces Cerevisiae. So it's a single-celled fungi. So it is a eukaryotic unicellular microbe. And so here's what humans have figured out, that ethanol, you know, if you leave grapes sitting around for long enough and, you know, you're hungry enough, when you go back to get those grapes, it looks like they're rotted, but you're starving. So you know what, I'm going to kind of eat that gross grapiness. And then all of a sudden you feel like you have superpowers. So that's probably how, you know, the whole wine industry thing got started, because people realized that this type of fermentation, it didn't kill you when you ate those rotten grapes. It actually gave you superpowers. And then of course you can now make it a little more tasty than what I just described to you. So you probably knew about wine. You probably knew beer also as a product of fermentation of the sugars that occur in grains in this case. So this is fruit juices, these are grains. You know, who doesn't want to see a drunk squirrel drinking beer? So I included that one as well. But did you know that fermentation is also an important thing that goes into making chocolate, and it goes into making coffee, because cocoa beans and coffee beans are broken down and fermented to take out the bitterness and to give that, you know, those other flavors that come along with coffee and chocolate. So you can't just take the beans and, you know, chop them up and make chocolate out of them. You actually have to put them aside, let them ferment biologically for a period of time, and then process what comes out the other end. And also I want to point out that bread rises as a result of an alcohol fermentation. So the CO2 that you see being liberated right there causes the bread to rise. And the bread rises because of wheat having a high content of this very stretchy protein called gluten. So what happens as the CO2 builds up, the gluten stretches and the bread rises. When you bake it, the alcohol evaporates and you end up with those fibers being solidified into bread. So we figured that out for a very long time. We've known how to make bread, we've known how to make wine, and we didn't know how it all happened until Louis Pasteur figured it out for us. But so there's that. And then looking this way, so here's pyruvic acid, and then this bacteria, Lactococcus and others, will complete that pathway by transferring back the hydrogens back to pyruvic acid, forming lactic acid. And as I mentioned, yogurts and cheese are formed this way. Fermenting lactose in the milk produces lactic acid, which causes milk proteins to curdle because the pH of the milk goes down quite a bit. And if you remember, of course, that proteins are denatured in under acidic conditions. So butter is produced as a result of that. Yogurt is produced as a result of that. Again, all things that we figured out as soon as we realized that other mammals, we could partake of their milk and use it as well. So then there's all this other stuff. Okay, so a couple things I want to point out. Here's a type of fermentation, which is called a 2,3-butanediol or butanediol fermentation. It's an alcohol fermentation. Butanediol is something that's being investigated as a potential additive to put into fuel in place of ethanol. And the reason for that is because ethanol is currently produced through the fermentation of corn. And when farmers are growing corn to put into gasoline, farmers are not growing food crops. So that has actually become a problem. So they're looking for bacteria and other things that will do this fermentation using garbagey stuff. Okay, so in our area, wood pulp that goes into the paper industry is just so you know, as the wood is being processed, the cellulose is what goes into making paper. So the pulp is waste. And so bacteria can actually get into that waste and ferment those leftover compounds, turn them into butanediol instead of growing corn and using the corn exclusively for that. So that's that's research, an area of research that is underway to try to come up with solutions to those problems. Looking down this way, here's another pathway leading to acetone, which is a chemical solvent, down to isopropanol, which is rubbing alcohol. So that's rubbing alcohol, not to be drank at all, is also produced as a result of bacterial fermentation. Butanol is another one of those industrial solvents or that occurs for these types of pathways. So then over here is something called the mixed acid fermentation pathway done by a variety of different types of bacteria. And you'll notice there's a couple ways to get to it. So we can go this way or we can go that way. But the point being, our pyruvic acid or the byproduct here is actually converted into, look at all these different acid compounds. Oxaloacetic acid, malic acid, fumaric acid, which some of those names you might recognize. Succinic acid. And the point being, these are very acidic compounds. So the end products are very acidic. This is called mixed acid fermentation, as opposed to the lactic acid fermentation, where only lactic acid is produced. There's a variety of different acids that are produced. And here's what we've learned to do with that. So generally, when bacteria ferment sugars, they turn it into acids. All right, so something like acetic acid, which you can see over here. Acetic acid is vinegar to varying degrees. Here, these acids are a little bit less acidic-y. And so these are used for fermentations of vegetables like pickles and whatnot. So when you pickle something, you are actually using a fermentive process, not when you ferment it, you are. When you pickle it, you're just putting the vegetable in the acid already produced. When you pickle it, you add the vegetable to a solution that contains sugar, and the bacteria that are doing this fermentation, they produce the acids. The acids preserve the foods. So this was a discovery that changed humanity because it allowed us to store foods before the days of refrigeration. We had been canning and storing foods by allowing bacteria to go through this process and produce it for us. So obviously, because there are different populations of bacteria that do these things, like the wine industry, why Pasteur became famous and why he was doing what he was doing was the wine industry was trying to figure out why sometimes they got great wine, everything was wonderful, but every once in a while their wine came out a little, you know what I mean? It came out a little acidic, nobody liked it, and they were losing money. And that's when Pasteur stepped in and figured out all these pathways. And he found out that when you had yeast and not bacteria, you got ethanol. When bacteria were involved, you got acids. And he came up with the process of pasteurization to remove the bacteria from the stuff you started with the fermentation. So you just added the yeast without the bacteria, you got great wine every single time. And so from that, the rest is history, as we put it. So that's the takeaway for fermentation. I also want to point out, because there are discrete chemical end products to these things, these are things we can detect in the laboratory, and we are able to use those as a basis for biochemical tests, which is, we've already started doing that, right? So for example, the TSI test, we're looking for glucose, lactose, and sucrose fermentation. We're looking for the end products. We're looking for acidic end products by measuring the pH. So not the specific thing, but we're measuring the pH. So and next week too, you'll see that as well. Okay, so I guess what I did here was to highlight all of these things, which I forgot completely that I'd done, right? So here's the ethanol fermentation, and then all these other different types of fermentation. Should be one more. There you go, the mixed acid fermentation. Okay, so this is all done if there is an endless supply of sugar. The microorganisms can go through this process of fermentation. They can make enough ATP to get by. But what do you do if there isn't any sugar? So my first question is, what if there isn't any sugar? Can living things survive if there isn't any sugar? And you might be thinking, no, I would not be able to survive if there wasn't any sugar. But the truth is, we can survive if there isn't any sugar. So us complex organisms, we can actually take other stuff and make sugars out of them. And microorganisms can just take whatever organic stuff is available and do what they have to do with it. So that's where this idea that we can also metabolize proteins and fats, catabolize them, make ATP, get our metabolites, and feed them back into these other pathways, also that we can make ATP, but also realize that there's all of these different pathways lead into something that's referred to as the Krebs cycle. On my previous slide, I showed you that crazy thing with all the different types of metabolic pathway. Notice that the Krebs cycle, that circular thing in the middle, was in the middle of all of them, because the Krebs cycle truly is the transformative pathway in cells. it doesn't get the recognition that it deserves. So I want to give it some of the recognition it deserves, because it's really the ultimate energy and metabolite harvesting cycle that occurs in cells. So all of glycolysis feeds into it, the catabolism of fats, first breaking them down into their glycerol fatty acids, feeds into it, breaking proteins down to their metabolites, amino acids, they can feed into this. But ultimately, all of those things that are releasing the electrons and hydrogens are being passed around through this Krebs cycle. So we're going to do a close-up on the Krebs cycle. Don't be scared, because this looks crazy. This is the Krebs cycle. All right, so here comes the metabolites coming into the Krebs cycle. They are metabolized in this circular fashion. And again, focus on what goes in, what goes out. So lots of things go into this. And there are lots of ways you can plug into the Krebs cycle, not just from glycolysis, which is the way you learned it previously. But as long as this cycle goes on this way, what you want to realize is that stuff goes in, stuff goes out. What's the transformation of the matter and the energy? So the matter is just being transformed in the cyclical fashion between the 6 carbon compounds and 4 carbon compounds, and then we just keep going. What comes flying out? Energy, right? So the energy comes out in the form, again, of those electron carriers, also ATP. So there's our energy that's being transformed. But notice also we end up with what's left of the matter at this stage of the game, because the hydrogens in this matter are now affiliated here. CO2 is the waste. CO2 is the low energy stuff that is the matter. That's the transformation of matter right there. But then notice that coming out of all of these different metabolites, these intermediaries, if you will, are building blocks for biomolecules. So here's metabolites that are flying off of here that go into building nucleic acids. Here are metabolites that are used, fatty acids and sterol molecules that are used to make lipids out of. Here are where amino acids are the metabolites that get spit back out. So then those can go into biosynthesis or anabolic pathways. So all of our anabolic pathways, all of our taking those metabolites, adding energy and building them up into biomolecules, here's where all those building blocks are coming from. Okay, it's not all of them, but there's a lot that comes from the Krebs cycle. So I didn't want to just, go through the, oh yeah, energy, because that's what we always do, without giving a shout out to the Krebs cycle as the central pathway of metabolism. My opinion, but I think that of other biologists too, okay? Okay, so when we're done with this, our matter is transformed in carbon dioxide, which is a gas, which goes off to be part of the carbon cycle, right? We have all these different metabolites, and then we have the energy. So once again, we have NADH, and this is flavin adenine dinucleotide, which is holding on to the electrons and the energy. And remember what I told you about NADH, which is that it is, as a molecule, it's not very happy because it's unstable, and here is it's stuck holding the electrons. It wants to give them away. So the Krebs cycle energetically is also not complete. So how do cells complete the Krebs cycle? And the answer to that is through respiration, through electron transport phosphorylation. So let's start with some words just to kind of refresh your memory about it. So after the Krebs cycle, respiration, the 3 parts to respiration that you gotta consider: the electron transport system, right? So this is a membrane-associated series of redox enzymes in a chain where one redox enzyme wants the electrons more than the one before it, so they keep passing those electrons on down. The enzymes are located in a membrane. In your eukaryotic bias days, they told you the membrane was in a mitochondria. Now that you're here in microbiology, you know that mitochondria are actually bacteria, so you realize that they are in the cell membrane of the bacteria. The proton motive force is something that is generated thanks to the electron transport system. So this is how batteries are produced. The proton motive force pumps hydrogen protons across to create a gradient of protons. So you have an imbalance of positives to negatives, a pH imbalance, also an electrochemical imbalance, if you will, and that creates a potential energy. So that is the proton motive force, which would not get generated if it weren't for the electron transport system making electricity by letting electrons flow through the electron transport systems. So the proton motive force runs on the electricity generated by the ETS. And then, because we always have to have a third word, chemiosmosis. So chemiosmosis is that process where we take this energy and we store it in ATP. So it's going to catalyze the reaction that makes ATP by adding that last phosphate group onto it. So that's called chemiosmosis. Those three things are what we'll talk about finally. And I want to point out that this is the basis of fuel cell technology. So if you are interested in this sort of thing, a fuel cell is essentially a mitochondria or a bacterial cell, if you want to look at it that way. So you've got, hydrogen tanks. So instead of NADH and FADH having all these hydrogen protons that they're going to hand over for electron transport, what they do is they supply the hydrogen. So for a fuel cell to work, you have to have a supply of hydrogen, which, by the way, is one of the reasons why fuel cell technology in cars hasn't worked out so well, because you have to have your gas stations would have to have hydrogen pumps in addition to gas pumps. But anyway, so you have to have a supply of those hydrogen protons, and then the fuel cell stack is actually that process that is producing the electron transport. So this energy in a fuel cell is actually used to turn the wheels. That's where the energy goes. So in other words, it's not stored, it's used, it's transferred to making the wheels turn, making the car go forward or back. So one thing that's the big takeaway from all this is to just realize that prokaryotic cells, which were the predecessors of mitochondria and eukaryotic cells, were the inventors of this fuel cell technology. So physics and chemistry came first, biology jumped in and was able to take advantage of that. And then humans were like, oh, we could do that. And we created fuel cell technology. But it was, 4 billion years later, just saying. Okay, so here's, again, that bacteria are the ones that are actually doing that. So here is a representation of the electron transport system and proton motive force. So it's always kind of like, this is an animation, so I'm going to let it roll. And it's always like, where do you begin? I want to start with the electron transport system. So while this is rolling in the background, let's look at that. So what is it? It is a series of enzymes, redox enzymes. That's these. There's more than 3, but that's okay. So it is a series, it's a redox chain. So in other words, this enzyme right here wants electrons more than that one does. This one wanted the electrons so much that it was happy to grab them from NADH and FADH. Remember where they came from? They came from the Krebs cycle. Bringing those, they just keep bringing them down there because that's where the matter went and the energy. So there's our little cloud that we had from the Krebs cycle. So notice NADH is going to show up and it's going to drop off its hydrogens because it's catalyzed to do that. There's our hydrogens, which are split into protons. Let's follow the electrons. So the electrons flow through the redox chain. So this enzyme grabbed it from NADH. Now it's going to pass them on to this one. And now it's going to pass them on to this one. Then it's going to pass them on to this one. And then ultimately waiting here at the end is the final electron acceptor, which is oxygen. Here's the deal. This flowing of electrons through the electron transport system is electricity. The electricity is used to run these pumps. These are called proton pumps. They are in the membrane. There's lots and lots of uses in biology of proton pumps, by the way. But notice what happens. So going back to here, here's our NADH. So we remove the H's from them. We split the hydrogen atoms. The electrons are going that way. Look at what the hydrogen protons are doing. So here they get pumped to one side of the membrane. And then water dissociates, and these pumps keep pumping these hydrogen protons to one side of the membrane, thanks to the electricity that's being generated by the electrons that are flowing through the electron transport system. So when we have this imbalance of more positive charges, more negative charges, right, a higher concentration on one side than the other, that creates a gradient of potential. So this is a membrane potential. And when this is where the proton motive force comes from. So the final step of this is the chemiosmosis. So how does the energy that's being generated through this proton motive force, how does that get transferred into a usable molecule like ATP? So we're going to have to wait for our electrons to do their thing and our pumps to do their thing, creating the proton motive force. Here it is. As these hydrogen protons flow through this, which is called ATP synthase, the energy that this generates is enough to make this bond occur. So this enzyme called ATP synthase, which is actually a bunch of different proteins put together, bunch of enzymes put together in a very unique way, which means this thing spins like a turbine. So as the gas, the hydrogen flows through this way, the turbine spins, that mechanical energy is transferred to make the bond, which forms ATP. If that isn't brilliant, I don't know what is, right? So this is how biology invented fuel cell technology. We're still doing it same way 4 billion years later. That's pretty cool, right? So just watching it one last time as we go through this process, the ATP synthase, the proton motive force, chemiosmosis, meaning the transport of these hydrogen protons through this, creates the energy that makes that bond form. So there we go. And that completes respiration. So we complete by transferring the energy, right? So all the matter are already dealt with. So this is the final step in transferring the energy. The energy is transferred to ATP. And I will say that most cells also produce heat this way, but this is a very efficient energy process. So in a fuel cell car, you're losing, actually most of the energy is going to heat. In a mitochondria or a bacterial cell, most of the energy is actually going into ATP. So how much ATP, you might ask? And I also just want to say, we're going to look at that, but we're also going to realize that what I showed you was aerobic respiration, because oxygen was the final electron acceptor. Oxygen being the thing that wanted those electrons more than any of those redox enzymes, because oxygen really, really likes electrons. That's why oxygen, the word oxidation, comes from oxygen, which is the ultimate oxidizing agent. It wants to be reduced. So reducing oxygen gives us water. So bacteria live everywhere, and ones that live where there is no oxygen, the thing is, how can you live where there is no oxygen? And until the golden age of microbiology, it was presumed that life could not exist where there was no oxygen. However, here we see that there are bacteria that can use other electron acceptors that are not oxygen. So, and by the way, they're not as needy as oxygen is when it comes to electrons, so they're not as good at this as oxygen is, but they are pretty darn close. Okay, so here's iron can be used as a final electron acceptor in place of oxygen. Nitrate, nitrite, or other nitrogen gases. This is part of the nitrogen cycle, which you'll hear about. Sulfate can be used as an alternative to oxygen. Sulfur, this is tetrathionine, right? So these are all different sulfur-containing compounds. So if those chemicals are present, if you have a food source, an electron donor, and you have one of these things as an electron acceptor, and you are a bacteria that has it hardwired into your DNA to produce the enzymes that allow you to transfer those electrons to these alternative electron acceptors, you can survive anaerobically. And so we can look at that. Again, this is a trait. These are traits that we can measure in the lab by doing tests for anaerobic respiration. The nitrate reduction test is one, which you've done. We've done that this week. And the the triple sugar iron test, which again, my face is in the way here, I think. But the precipitate, the black precipitate in the TSI tube is H2S as a result of sulfur reduction or sulfate reduction. So that's a way for us to look at the bacteria, look at their traits, realize that these traits are hardwired into their DNA, so it's not part of their genome that they are able to do this, and identify them and actually learn more about them overall. So what I was saying about how oxygen, the process of transformation of energy is most efficient when oxygen is the final electron acceptor. But like I said, it ain't bad. It's close when you're using alternatives, right? So here's aerobic respiration, where oxygen is the final electron acceptor. And notice these are the yields of ATP molecules. Again, you get a little bit out of glycolysis, but most of it comes from the electron transport and chemiosmosis. So this is from 1 mole of ATP, which is not the same as a molecule, even though I think it should be, but I'm not a chemist, so I'm not going to argue with anybody. You can get 38 molecules of ATP. Remember, this is all cellular energy. This is gas in your car that can be used for doing cellular work. An anaerobic bacterium using one of these alternatives, you can get anywhere from 5 to 36 molecules of ATP. So again, not as efficient as if you were using oxygen as a final electron acceptor, just because they are not as needy for electrons as oxygen is. And then fermentation, all you get is the little bit of ATP that's produced during that first glycolysis phase. So it's a completion, but it's not very energetically efficient. So you're only making a little bit of ATP, and realize to make a living off of that, you have to live someplace where there is an endless supply of sugar to be able to make it worthwhile. Or you could use it as a backup pathway, which lots of bacteria do. So they'll use respiration of one form or another, and then they use fermentation just in case there happens to be any sugars that are available, just to kind of, get a little bit more energy out of the deal. So, and I also, even though in some of your eukaryotic bias courses, you may have heard fermentation being called anaerobic respiration, it is not respiration, because respiration requires ETS, the electron transport system, and chemiosmosis to be complete. So they're not the equivalent. It's an anaerobic pathway, meaning it doesn't require oxygen to make ATP, not very efficient, not anywhere near respiration, okay? So that's a little microbiology perspective on that. Okay, so hopefully the idea that we have anaerobic things living and they're doing chemistry that is foreign to anything you might have known about before isn't too disruptive. Because now I want to talk about something that is completely bacterial. Bacteria and archaea are the only ones that are able to do this, and it's referred to as lithotrophy. So litho is refers to rock. Trophic, remember, means to love. So these are rock lovers, right? Rock lovers. Trophic also can refer to eating, what things eat, right? So what is their lifestyle with refer to what types of electron donors they use for food. So lithotrophy, these are bacteria and archaea that eat rocks. So they're not starting with glucose, they're not starting with carbohydrates, lipids, or proteins. They're starting with inorganic minerals. And they are doing what we do to organic compounds, only starting with these inorganic compounds. So their food, if you will, is stuff like rocks. So I I want to share with you just because this is Yellowstone National Park. This is one of the hot springs there, which is where there are an abundance of these lithotrophic types of bacteria that are living by metabolizing the minerals that the hot water are liberating from the rocks that are in the ground underneath. Okay, so just for a second, we'll just all take a second and go to Yellowstone National Park and watch the geyser and realize there are bacteria in there that are doing metabolism, and you wouldn't think so because that spring is so hot, but I love that. Okay, so there, we feel nice and peaceful and calm now. So they're using as electron donors inorganic substrates. That's why they're called lithotrophic rock eaters. They can use oxygen as a final electron acceptor, but guess what? Lots of them are anaerobic, like the ones that you would find here. Could be anaerobic to complete the electron transport system and respiration also. But here's the deal. So lithotrophs, so this is how they're making ATP. So they're oxidizing inorganic substrates and using it to make ATP. They still need a supply of metabolites, of biomolecules to build their cells from. So that has to be supplied in other ways. And a lot of them do it by making their own organic compounds, meaning they do something equivalent to photosynthesis. And others just, so they make ATP by eating rocks, and then they go over and they will also use organic compounds from pre-existing things like we do. So they are really interesting, cool, awesome bacteria. One of the most famous microbiologists in recent history was Thomas Brock, who was the first guy to say, you know what, there's bacteria in these springs, to which lots of people said, are you crazy? That's 120, 30, 40 degrees in there. There's no way bacteria, nothing can live in those springs. That's why they're so beautifully, perfectly clear. He said, nope, I'm telling you. And he found out, guess what? There were bacteria present and discovered this whole idea of lithotrophy. So let's look at a more pertinent example of lithotrophy. Okay, so a discussion of the nitrogen cycle, which is something that we're also looking at in lab. Because the nitrogen cycle, this is an earth cycle, an ecological cycle. It is a movement of nitrogen from forms that are not usable to living things into forms that are. So the forms that are generally are nitrate and nitrite, ammonia, right? These are things that can be incorporated or fixed into organic compounds that we can use as biomolecules or metabolites. And then there are those that do this conversion into these are unusable forms to biology, right? Obviously they're important to the outside world, but we can't use them as building blocks. We can't put them into our biomolecules, but they're necessary because we need them to get back to the forms that are. So organic nitrogen, inorganic nitrogen, and it's this giant cycle that occurs, and we've studied it in great depth out in the other world, and now we realize that these, in many ecosystems there are examples of nitrogen cycles as well. Guess what? You individually are an example of just one of those mini ecosystems, if you will. Okay, so the cycle is just looking at these up here. Okay, so this is up here is lithotrophy. So the nitrifiers, which are the ones that are adding organic nitrogen or forms that could be used for organic nitrogen back into the environment, those nitrifying bacteria are oxidizing ammonia. By oxidizing, I mean they're using it as electron, they're eating it, right? Electron donors to be transferred through the electron transport system, making ATP. And when that happens, when ammonia is oxidized or used as food, you get nitrite and then you get nitrate. Okay, so these are usable forms. Then, on the other hand, there are the denitrifying bacteria. These are using organic compounds as food, but they're using these nitrogen-containing gases as final electron acceptors. So this is anaerobic respiration. Lithotrophy Anaerobic respiration, right? So these are microbes called heterotrophs that are doing that because they're eating organic stuff. They're just using these alternatives to oxygen. These guys are actually eating the ammonia as their food, and as a result of that oxidation, they're producing these usable or nitrifying types of compounds. So we have a way, we are testing for these things. In lab, we can test for nitrate conversion to nitrite and then denitrification using the nitrate reduction test. We'll leave that for lab. I want to point out, like I said, you are one of those ecosystems where we're now finding that nitrogen cycles occur. So here's an example of us as we have become more familiar with our microbiomes, the fact that we are inhabited by all different kinds of bacteria. We're also being more conscious of the fact that there is an internal nitrogen cycle occurring, which is a recycling of all these different nitrate or nitrogen forms so that we can have a supply of them for our own biomolecule production. So notice, right, the nitrate reductase, nitrate to nitrite reduction is when nitrate is used as a final electron acceptor in place of oxygen, anaerobic respiration. That gives us nitrite, right? So nitrite can also be used as a final electron acceptor. Here we're giving credit to the gastric mucosa, but I'm also going to give credit to the gastric, the microbes that are in your digestive tract as well. And ultimately, they produce this stuff called nitric oxide. So if you're in healthcare field, maybe you recognize nitric oxide because it is known for its health. It does good things for us. So most recently, it decreases blood pressure, it decreases inflammation, causes vasodilation. Guys, anybody that might be using Viagra, this is what happens with Viagra, right? So you stimulate vasodilation by triggering nitric oxide. So it also has its, it can have a dark side. So you got to be careful because pathways, you think, oh, well, this good stuff is happening, but guess what? Too much of a good thing can also cause problems. So the nitric, and one other thing I want to say is an article, right, that article that I just linked to really briefly in the schedule shows that they're using nitric oxide to decrease inflammation in the lungs of COVID patients, right? So patients that are having that acute episodes with inflammation, and that includes cardiovascular, by the way, the nitric oxide is calming that down. So that was a study that was published literally, just a couple of weeks ago. So, again, another shout out to our microbiota, which are contributing to this process inside of us, right? So we got to give them the building blocks that they need to be able to do it. So that's the nitrogen pathways, right? Lithotrophy, and then also heterotrophy. And then looking at phototrophic metabolism, all right? So what does that mean? Light loving. So these are pathways that you've heard about before, but we're gonna just kind of talk about it from the perspective of bacteria and other microscopic organisms that do it. Algae do it, right? Trees do it. You've heard enough about trees and photosynthesis in your lifetime, so I'm gonna just stay away from that. Algae are phototrophic. Many types of bacteria are phototrophic, which means they can absorb light energy and they convert it into cellular energy, ATP. They can do that conversion using the proton motive force and electron transport. So similar to respiration, right? Redox chain, produce ATP, but they convert that light energy by absorbing it and then using it to activate or increase the energy of electrons. So that is where the ATP comes from, and then realize that this is an anabolic pathway, the Calvin cycle. So this is the catabolic side. On the anabolic side is this Calvin cycle, which is another recycling process, which recycles organic compounds, and it ultimately results in the production of organic carbon from inorganic carbon. And the only form of inorganic carbon that you need to worry about is CO2, right? So CO2, CO2 No hydrogens in there, so that's considered inorganic carbon. It's very low in energy because it doesn't have any reactions left to do. So this is referred to as carbon fixation. So CO2, what happens is the energy is transferred back to the CO2, and ultimately what you end up with is our sugar molecules, right? Small sugars, and then they can get larger, and then those are the metabolites that go off into the construction of, you know, carbohydrates and actually glycoproteins and glycolipids and anything that has sugar molecules in it, okay? So phototrophic refers to the fact that the energy is coming from this photo process, this process of converting light energy into ATP, which that ATP is then used for carbon fixation. So that, if you remember, is the basis for this big web called the carbon cycle, which you probably remember from your previous biology days. So what I want you to realize is that the carbon cycle, you were only given half of the story when you heard that in general biology, because there is the aerobic version of the carbon cycle, which is the one you heard about. You know, that's the one that always has the flowers and the trees. And then, you know, there's always a cow or a puppy or a kitten or something to show how carbon flows from CO2 into organic compounds, and then those organic compounds are respired in mitochondria from the puppies and the kittens, and they breathe out the CO2, which is then used by the pretty flowers to fix it back into using the Calvin cycle, right? Phototrophic metabolism. And right, this is all you knew up until just now. But I'm here to tell you about the dark web of carbon. This is the dark web of carbon over here. This is light independent, it is anaerobic, and it is only done by, guess who? Bacteria and archaea. So by the way, this is a book, and I don't know anything about this book. I'm not advertising the book. I just like this because it summed up what I'm trying to say here. It's the dark web, the dark, quiet web. Don't tell anybody about it. But this is an essential part of, you know, the recycling of carbon in our environment. It's often just like forgotten and neglected, right? When we talk about, you know, carbon and CO2 and global warming, guys, we got to pay attention to this too, because guess what? The microscopic organisms doing the dark web outnumber the macroscopic ones doing this side of the pathway by a huge amount. So, you know, just kind of looking at this without too much ado, it's bacteria and archaea, but the one that I want to talk about, methanogenesis, methane genesis, methane producing, is archaea. These are archaea, populations of archaea, which are doing this dark web, right? So the organic matter gets broken down, releasing CO2. And notice when the final electron acceptor is carbon dioxide, you end up with the product of CH4, which is methane. So methane is the end product, if you will. So, right, when you're doing it with aerobic respiration, the end product is water. When CO2 is the final electron acceptor, you get methane. And so the supplies of methane, natural gas in the ground, are done as a result of this dark web of carbon, if you will. And a lot of other stuff too, by the way, which I won't even bother to go with, just methanogenesis being an important part of that whole thing. Okay, so take home, it's a bacteria and the archaea that are the ones doing this. And they deserve equal time when we talk about carbon, so I'm giving it to them because that's what I do. All right, so let's put this all together in one place at one time and wrap this up. So based on their metabolism, right? So remember, their metabolism, bacterial metabolism, cellular metabolism is based on what is in the cell's genome, what's in their DNA, what genes do they have. They have genes for certain enzymes that allow them to do these chemical reactions. Do they have the gene for cytochrome oxidase, which says they can hand electrons to oxygen? Do they have genes for nitrate reductase, which says, nope, I'm not handing it to oxygen, we're giving them to nitrate. So all that's in their DNA, just like it's in our DNA. And then when those genes are expressed and they do metabolism, we can categorize all living things in these basic categories. So we look at, in terms of I kind of call these the lifestyle. How are they going about this process? What do they start with? How do they go about this process to transform biomass using metabolism? So we can look at it based on, all right, what are they using for their energy or electron donors? Or what are they using for the carbon to put into to build their biomolecules out of? Okay, so there's kind of like those two branches. So phototrophs are the ones that use light energy. So they use that light energy to energize electrons so that they can make ATP. Chemotrophs are the ones that are not, they're going to be using chemical energy, right? So this is light energy to start that process. These guys are using chemical energy. The ones you know the best, because we are one of these, are the chemoorganotrophs. These are also ones that are using biomolecules, breaking them down, releasing the ATP from them. We are this, right? So this is what our mitochondria do. So that we are using organic forms as the electron donors, and then, a variety of electron acceptors. Lithotrophs, the chemolithotrophs, are the ones that use the organic compounds for this, the organic minerals, as their source of energy. But remember, for every source you can't just have energy, you also has to have carbon that can go into building biomolecules. And so that's where these terms come in. Autotrophs, the carbon that is being put into the organic compounds that they build their cells out of comes from CO2. So autotrophs make their own food, meaning they're making their own organic compounds through that process of the Calvin cycle. Heterotrophs, on the other hand, I view heterotrophs, this is the laziest bunch of organisms that there are, because it's just like, I don't want to cook dinner, I just want to go out to eat. So it all comes pre-made to them in the form of organic biomolecules. So they'll break down those biomolecules, they'll get the building blocks they need, they get the energy that they need. Heterotrophs, that's like us, right? So it's sort of like, why make it myself? Why do all that work if I can just, go eat something else that has already done it for me? And that's what we heterotrophs do. So these are the categories that I'd kind of like you to consider. There's other ways of putting them together, but it's, where does the energy come from? And think about what that energy is used for in the end, right? What do cells use ATP for? Obviously to make more cells, but then how are they getting the carbon that they use to construct those cells out of too? The autotrophs versus the heterotrophs. So I hope, given that there was so many other options of lectures out there for you to look at, I thought hopefully this summed up what you needed to know for bacterial metabolism. So there's, between the lecture and me, I'm hoping that you have a good idea of how bacteria transform matter and energy, all the different ways that they do it. And again, realize that these are characteristics when we look at the products of these different pathways that allow us to learn more about bacteria, including discriminate between them. Who's doing what is about what's in their genes, and we can actually measure that in the laboratory. Okay, so end of story. Take lots of breaks if you need to, and I will see you in lab. Bye.